Welcome to the final Facebook Live for Brain Discovery Week. Today we have come to the Wellcome Centre for the Integrative Neuroimaging to conduct a live experiment, the first ever broadcast live of an MRI scan from the University of Oxford. Please send in your comments and questions and we will try and get these answered live within the interview. To help with the process, I am joined by Stuart Clare, brain scanning expert, and Holly Bridge, neuroscientist. Thank you both for joining us. Firstly, Stuart, can you tell us why we're here with a brain MRI scanner and what the competition was?
Yeah, so this is a brain imaging centre here. We use MRI to study the human brain. And right now the scanner is running the very first experiment, which is the winning entry from the Big Brain competition that we ran last year at the Museum of Natural History here in Oxford. So we had over 700 people enter this competition to answer the question, what would you do if you had access to this scanner to take pictures of the brain? What do you want to know about the brain? So we had just an amazing response. It was just brilliant reading all the entries.
And, Holly, how did you actually choose the winner?
Right, so that was obviously a big job. So from these 700 entries, we took a day and we got different groups of people to have a look through. And from those original 700, we whittled it down to 100. And then we got our panel of experts to have a look through all of those. And we came up with the top 25. And they were divided up. So we have our 10 from primary school age children. Some of those winners are here with us. We had our main experiment winners, so we have our winner is also in the room today, and then we had some that we thought were actually just too difficult to do in the scanner. So a whole variety of different types of winners, but it's great to have those winners here to see it performed live.
Yeah, and you mentioned the winner, who was actually a gentleman called Richard, who we're going to talk to a bit later on. But Stuart, can I ask, why did you choose Richard's idea?
Yeah, well, so Richard had this great idea to do with investigating how we process voices in the brain. And he was quite detailed about the experiments that he thought we could do, and he thought that we could not only look at how the brain was processing voices, but also what would happen if they were processing the voices, if they were distorted or speeded up in some way. And also what would happen if you saw a picture of someone speaking, but you heard a different voice. How would the brain cope with that mismatch? And all the judges just felt this was a really interesting idea, had really interesting elements of things that we could experiment with in our scanner. So we've programmed all that up and we're running it here today.
And I just had the lovely thumbs up to say that actually we've got some really exciting things to show you. But Holly, we are doing 3 experiments today. I mean, can you tell us quickly what the 3 are and maybe some results if they're coming through as we can see them?
Yeah, so we are running a series of 3 experiments to investigate this question. So obviously this idea of faces and voices involves 2 of our sensory systems. So we look at the faces using our visual system, and obviously we hear the voices. So in the first experiment, what we're actually doing is we're presenting pictures of faces and houses. The idea being that we can look at the activity to faces, where is the brain very active to pictures of faces. And we know that there are areas that are specialised in the brain that will be very active at that point. And in the next few seconds, we'll be able to show you some pictures. The second experiment that we're going to do our subject will be listening to familiar voices. So in fact, what we've done is we've been around our centre, we've recorded the voices and the faces of people within our centre. And actually, Stuart can tell you about an exciting thing that allows you to actually do this at home.
Yeah, so you can actually go online to the Oxford Sparks website and you can try this experiment. You can learn the voices of some of the people here in the lab as you watch their faces as well, and then you can see whether you can recognise them and think what's going on in your brain as you're doing that experiment. So when we're done the broadcast here, go off and try that on the website.
And we'll actually put that in the comments box below for you to be able to try that afterwards. So are we, what's actually happening right now, Holly, in terms of...
So if we go over to the picture of the brain, so what you can see in the background, so the grey structure, so if you look around the outside, we can see our skull, and inside there is the brain tissue. And on top of that grey area are these spots of activity which are shown in the red. The more active they are, the more yellow they become. So you can see that some of these are red, and then in the middle of those red areas we've got these yellow real hot spots. So there's a range of different areas, and we know that those are areas that are involved in processing of faces in the brain. So it looks like when our participant is looking at those faces, we can see that activity coming up. You can see that those red blobs that are being pointed at the moment.
So this is the results from the first experiment. So we're going to let now the guys do the experiment too, and we're going to talk a bit more about the MRI and how it kind of works. So Stuart, I've got to ask you, we keep mentioning an MRI scanner, but what is that, and how does it actually work?
Yeah, so an MRI scanner uses an incredibly strong magnet, and it uses radio waves to take pictures inside the human body. So just to give you an idea about how strong a magnet we mean, if you went to a scrapyard and you saw one of those big magnets that would pick up a car, that is 1 Tesla strong. So we measure how strong magnets are in units of Tesla. So that 1 to pick up a car is 1 Tesla. This magnet here is 7 Tesla strong. I mean, it'll pick up a double-decker bus. It's incredibly strong. And we use that in combination with radio waves to take pictures inside the brain.
And is that the reason that we're sat this side of the glass, would you say?
Yeah, so we want to make sure we're not getting too close to this very strong magnet. If you had some coins in your pocket or your keys were still on you, they would get sucked out by the magnet and attach, and we'd never get them off. And also maybe if you'd had an operation or surgery or something that had left something in you that might be ferromagnetic, we'd want to be careful that that wasn't being attracted in any way and causing any damage. So we check everyone before they go through into the room to be scanned.
And Holly, I have to say, we're seeing the feet of Aislinn and our participants, but
Yep.
What is it actually like to go inside the scanner?
Well, so as you say, we can only see her feet, and that's because essentially it's a long tube. So it's a tube about sort of 60 centimetres diameter. It's quite comfortable. She's lying down. She's fairly comfortable. And I guess the main issue is the fact that the noise that you can hear. So it is a very noisy environment to be in. She's got protection over her ears to reduce that a little bit, but it is that constant noise that you can hear.
Okay, and Stuart, I have to ask, is it safe?
Is it safe? It's a really good question because we can be concerned about scanners like this, but it is completely safe. Unlike X-rays or CT or anything like that, which uses ionising radiation, which can sometimes cause some small damage to cells, the MRI has no damage at all to cells, provided we're really careful that no one has anything that's ferromagnetic in them. So we screen them carefully to make sure there's nothing that could get attracted by the magnet, and if that's the case, we're safe to scan them.
And Holly, I think we might be able to see the results from maybe experiment 2. Do you want to talk us through it?
Yes, so if you remember for the previous experiment, most of those red blobs were at the back of the brain. But now, if we have a look at the brain, you can see now it's in the middle of the brain, and these are our auditory areas. So these are the bits of the brain that will respond when we're listening to things. And what she's actually listening to is these voices. We can probably get a clip of those coming through at some point. But you can see it's very, very active. Remember I said that the more yellow or white it is, the more active it becomes. So when she's listening to these familiar voices, she's heard them before, we get lots and lots of auditory activity. There's also areas that are specialised for voices. But one of the questions that we're interested in is, do you also get activity in the visual system? So if you hear a voice that you recognise, does that trigger some sort of activity in your visual system? So we expect that to be much, much weaker. So we wouldn't expect it to be that bright yellow blob showing lots of activity, but actually just little bits of activity. So that's what we're looking for at the moment. So I should point out that what we're doing, the amount of data that we're collecting is very, very small. And if we'd done this 5 years ago, we wouldn't have seen anything like this. So the fact that we can do this live on air is quite phenomenal, even for us. So at the moment we don't have any. At the moment we don't have our visual information.
And also, can I say, is there an opportunity to hear what Aislinn's hearing? Is there an opportunity to?
So one of the technical things not going quite right here. So it should be playing through my speaker here, but I can't get it to play right now.
That's
but
fine.
we know she is hearing it because we can see that auditory activation, the hearing activation in her brain. So we know she's hearing it okay, even though my speaker here has died for the moment.
So while we're waiting for maybe that visual part of the brain to come up, Stuart, can I quickly ask again?
Yeah.
Why? How is this scanner different to other MRI scanners across the hospital? Yeah,
so maybe you've had a scan or family members who've had an MRI scan in a hospital, and most hospital MRI scanners are 1.5 Tesla strong. This one is 7 Tesla. It's one of about 40 in the world of this really strong strength of magnet, and this is, we're using it for research because it's so powerful, and we hope that by taking these pictures in such a quick way and in such a fine detail, we're going to tell even more about how the brain works and also what happens when it's damaged and goes wrong.
And Holly, I got to ask, what kind of things do we usually get people to do in the scanner?
So that will be that will depend on your research questions. So part of the reason I'm here is my research area is the visual system. But the types of things that I do is looking at what happens when things go wrong in the visual system. For example, we might do something similar to this, where we play voices, because I look at people who were born without eyes, so they're blind, and what we want to know is what do all those areas at the back of the brain do if they don't get any visual information. Usually they process vision, but if they don't have that visual information, what do they do instead? And the answer is they actually do hearing and they do language processing. So this sort of experiment would be the sort that would excite the visual parts of their brain.
Which is incredible. So do we see different, I mean, I know we're waiting for the visual opportunities to come up on the brain, but do you see different areas being highlighted? Is that how we can identify what's happening?
So the brain is very specialised. That was the idea that when we did the visual faces, we saw activity at the back of the brain, and if we do the hearing experiment, it's actually at the side of the brain. If you get people to do more complex tasks like making decisions, then you tend to see activity at the front. So we have this specialisation of function, which allows us to predict where we're going to get different bits of activation depending on the task that we give.
Okay, and we've actually had a question come in, and I don't know whether this is something you can answer, but someone said that they think we could also identify what a stranger's voice might sound like based on some research that's been done. Is that right?
That's a very good question.
Just to put you on the spot, obviously.
So we have confirmation from our language expert that is true. We would make a prediction, and actually that relates very nicely to this third experiment that's running, because in this third experiment what we've done is we've taken some unfamiliar voices and we've paired them with the photos of the people from our centre. So what our participant will see now is faces that she knows, but with voices that she's never heard before. So what we're going to try and look at is how the brain activates to that compared to when they are matched up. So clearly there, if she's making a prediction about what people should look like from their voice, it's probably going to be wrong.
Yeah.
So we'll see. We might see some areas of activation that might answer that question.
And someone's asked a question saying, you know, what exactly lights up in the MRI? You know, what is the white and what is the red?
Yeah. Okay. So yeah, that's a really good question, because what we're doing is we're taking a sort of movie of what's going on in the brain. So every 3 seconds, we're taking a fresh picture of the brain. And as the part of the brain is working really hard, so maybe when we're listening to those sounds, the auditory parts of the brain, the hearing parts of the brain are going to be working really hard. They need a whole load of oxygen, and that oxygen is provided in the blood that starts flowing into those areas. And in this movie that we're taking, we can see the changes in the blood that are flowing to that area. And so we highlight that in red, where we're seeing any changes that are occurring. And that's why it's really important that we look at the difference between doing something fairly boring, like just laying in the scanner and it's, you know, listening to no sounds, compared to when we're getting them to listen to voices that they recognise, because it's the difference between when they're just lying there to when they're listening to the voices that we're going to pick out and highlight in red on the pictures here.
So while that's experiment is running and we're waiting for the results, I have to ask Holly, can the scanner tell what you're thinking?
That's what everyone wants to know. So yes and no. What we do is we look. The scanner can give us patterns of activity. So as you do something, like if I were thinking about someone's face, I was thinking about someone I knew. I know as a vision scientist that the pattern of activity I get would include some of those face areas that we saw earlier. So if I said to you, if I asked you to think about either a pub or your partner's face, I would be able to tell which one you were thinking about from the pattern of brain activity. So we can do very simple things like that. But of course, our thinking generally is much more complex than that. And we don't really have the capacity to look at the level that we need to look at in order to actually know what you were thinking. So we can do simple things, yes, but generally no.
And Stuart, I've got to bring it back to the whole point that we're here is this amazing competition that we ran for the Brain Diaries. Unfortunately you can't see, but we've got a packed room full here. Everyone's kind of stood watching this experiment. I was going to ask, there were some great questions about twins and things like that. Do you want to talk about a couple of those questions?
Yeah, we had some brilliant questions that we just weren't able to do. But one of the winners who's in the room here today said that they're a twin, and one of a twin. What would happen in their brain if they looked at a photo of their twin or if they looked at a photo of themselves. And we just thought it was fascinating that they would look very, very similar, and you would see that the picture is almost the same. The brain would have maybe processed the vision in almost the same way, but you'd know that one was yourself and one wasn't. And we just thought it was a really interesting idea. How does the brain activity look different from looking at the twin to you looking at yourself? So that was one of the brilliant ideas that we had.
Brilliant. And if you want to have a look at all the kind of runner-up questions, please go to the Oxford Sparks website. They're all on there, and you can kind of see what the judges thought of those as well. But Holly, I have to kind of jump back in. I think we're really excited to reveal actually the experiment. So do you want to talk us through what we're seeing?
Yes. So you can see now there are red blobs in various areas of the brain. So we're going to start with the bit that's been pointed out now. So that is the anterior cingulate area of the brain. And so these are the areas that are more active when she's seen the voices matched to the faces. So it's what she was expecting, as opposed to when they were mismatched. So it looks like that's involved in noticing those correct faces and voices. We also have, I think we've seen in both the auditory system, in the visual system, there's some red blobs back there. Those are the visual areas of the brain. And I think we can also go to the auditory. In the auditory areas, there was also some red areas, which suggests that actually what's happening is that when you're getting information that corresponds in both your auditory and your visual system, you get a boost in your activity. Because both in the auditory and the visual system, you can get an increase in activity when the voices and faces are matched compared to when they're not.
So can you talk us through a little bit more, like, different areas of the brain? I mean, do they highlight things like emotions and memory and things like that?
That's all part of this specialisation of function. The different areas of the brain all have a specialised function. So we've shown the visual areas and the auditory areas. So they're the sensory areas. But of course, there are those more complex bits of our brain, like, as you say, the emotion, where it's hard to here we could show a picture of a face and we can get activity. But to pick someone in to look at the emotional areas of the brain is much more difficult because you can't get someone in and get them to immediately feel an emotion. We might do it by using scary faces or showing pictures of sad people, but it's not as direct a measure of those areas. But yes, we know that all those different brain areas have these very, very different functions.
And I was going to ask you, obviously we've just kind of seen it, so can you kind of sum up, the answer to kind of Richard's question that he kind of, put for the competition?
Sure. So I guess his question was, what happens if you interrupt or if you sort of mismatch these faces and the voices? And I think what we can see is you can detect that by looking at the patterns of brain activity. So your brain does respond differently depending on whether or not you can match your face and your voice. And clearly a lot of that is to do with your expectation.
And like I said, you can do the experiment yourself at home. Look at the comments box below, and we have put that. Someone's actually asked, are there areas concerned with speech and language plastic? Is that plastic? Yes, plastic.
So plastic is the word that we use to describe the brain's ability to change over time. And we now know that throughout our life, our brains change. And of course, these language areas must be plastic because we can all learn new languages, irrespective of the age that we are. As youngsters, we learn them very quickly. The brain is very plastic. As we get older, it might take us a little bit longer and we might not remember as much of it. But we can still do it, which suggests that the brain has to be able to make these changes. It has to be plastic.
Yeah, absolutely. And Holly, I'm just going to give you a break and ask you to swap seats actually with Richard, who is actually our competition winner. So Richard, lovely to meet you and be part of this experiment. Can I just say, firstly, did you ever think you writing your little kind of question on a bit of paper would get you here today?
Absolutely not, no. I actually came to Oxford that day on my birthday. And it was a lovely day, and we went to the exhibition, fascinating exhibition, and thought, well, I love entering competitions anyway. I've done a few over the years. I thought, well, I'll give it a go, and just sort of came up with that idea. But it was something that actually has always sort of, I've always thought it's a wonderful thing for the human brain to be able to pick out a voice from within a crowd of thousands of people. If they're all shouting, you'll probably be able to home in on the sound that you're looking for. Firstly, how does it do that? So it's obviously sort of picking up on a signature to the voice. But what I thought, well, let's play with that a little bit. Let's see whether the voice, if it's then distorted, whether the brain can still find a key element of that signature, which then means it can identify. And I thought that would be something quite interesting to look at.
And how have you found watching the results?
This has been, I just feel so honoured, and it's just been such a fantastic experience. It feels like we're in a submersible at the bottom of the Marianas Trench, exploring these wonderful areas that we know so little about. They're right at the forefront of neuroscience here. And looking at that brain, I can hardly believe I'm actually looking at the brain inside a cranium of a lady in that MRI scanner. And it is just superb, because 10, 20 years ago, that wouldn't have been able to be done. It was only invented about 15 or so years ago. And I think with the power of magnets, we've now been able to look into something that people have asked questions about for millennia. So yeah, fascinating.
Yeah, and I have to say, Stuart, just going back to you, like how many scans do we actually do a day here in Oxford?
Well, in this centre here, there's probably 12, 15 scans a day in this one centre, but there are lots of other MRI scanners around, all scanning for research, trying to find out more about the brain and body.
And someone's actually asked, Stuart, what made you actually choose to go into neuroscience and brain imaging?
Yeah, I mean, it was really, it's a brilliant area to be in. So I'm actually a physicist by training, and so I studied physics in university, but I was really lucky to study in Nottingham, where a lot of MRI was invented and developed, and so I got the chance to work on some of the very first of these functional MRI scans that were ever done in the country, and so that got me into this way of using technology to study the human body. And actually, I mean, I don't even have a GCSE in biology. I was never really interested in biology at school, but having learnt the physics and then come into it through the technology kind of route, I've just then got fascinated by the brain and everything about neuroscience. And it's just such a privilege to be working with everyone that I work with, the brilliant team I work with here, and just finding out absolutely fascinating things about the brain.
And we do tend to have a young audience, and we've got some really young audience in the crowd today. Like you said, it doesn't have to. You don't have to be, brilliant at science. You can have a passion with maybe computers. That's what's wonderful about the brain, isn't it?
Absolutely. Yeah. Modern science takes massive teams. We're such a big team here, and we couldn't do it. You can't just be an expert in just one thing here. You have to work as a team. You bring your expertise. So without the experts in statistics and computer programming or technology or medicine or psychology, we couldn't work together to do this. And I just love being part of the team, and everyone's got something to bring to that team. So even if you won't naturally think, oh, I'm a really sciencey person, maybe there's something in another area that would bring you into neuroscience.
And what I wanted to say, and probably the final question of the day is, the brain is still so under researched, and there's still so many fascinating things that we can find. Where do you think maybe the research can go? Well,
yeah, I mean, we're making so many discoveries all the time, and developing the modern technology is really revolutionising the way we study the brain, and also the way that we can treat and affect the brain. So it's just such a privilege to working at this real forefront of science. And I think over the next 10, 20 years, we're going to see some major developments and breakthroughs in our understanding of neuroscience, and hopefully treating neurological and psychiatric conditions.
Well, thank you very much for all of those that have tuned in today to watch this live, and to those that are in the room today. And thank you, Stuart, and the team for actually making this happen. It's been absolutely fascinating. Please keep your comments coming in. We'll pass those on to Stuart and Holly if we haven't covered everything in the live feed. And thank you for tuning in all week. We've had some great Facebook Lives, so please go back and watch about pain and how the brain copes with that. How does exercise make us have our brains work better, and how alcohol affects the brain. And we also looked at brain-based treatments for depression as well. So please visit Oxford Sparks to see all those again. And once again, thank you very much. And Richard, thank you very much for asking the questions today.
Yeah, you're welcome.
Thank you.